Hey everyone, Mrs. Walker here. This is going to be the video for the Meiosis Gizmo. However, I do want to tell you that if this does work for you, do not watch the video. If you can get into the Gizmo, do not watch the video. Please do the Gizmo yourself because it's going to be interactive and it's going to help you. But I wanted to put this video up so you guys were able to get into the Gizmo. So this is what the worksheet looks like. There are a lot of pages, but don't panic. It's just because the directions are real spaced out. There's not a lot to do. So up here at the very top, your vocabulary to be familiar with. You have seen these before, so definitely pay attention to your vocabulary. If you know the vocabulary, this process is going to be a lot easier for you. So they do have two prior knowledge questions here. Please do those before the gizmo. Do those before, you can pause the video at any time. So pause now and do these two questions. So then the warm-up here. So meiosis is a type of cell division that results in 4 daughter cells with half as many chromosomes as the parent cell. Remember, we go from diploid to haploid in meiosis. These daughter cells mature into gametes or sex cells. In the meiosis gizmo, you will learn the steps in meiosis and in experiment to produce customized sex cells and offspring. So this is what the gizmo looks like. So then we're going to get started. On the steps tab, click male. You're looking at a germ cell or a cell that will undergo meiosis to become gametes. So it says read the description of interphase at the bottom of the gizmo. What happens to the cell at the beginning of interphase? So steps, we're doing male first. Here is that information. So you can pause it and answer your question. Click on the DNA in the nucleus, describes what happened. I'm going to click. So I clicked on the DNA, and here's some information here for you to read. Again, pause this video throughout. Why is it necessary for the cell to grow and duplicate its DNA before the start of meiosis? So those 3 questions you can answer right there. In activity A, make sure that the steps tab is selected. If necessary, choose the male cell, click on the DNA to copy it, and then proceed to prophase 1. Unlike mitosis, which produces 2 identical daughter cells from one parent cell, meiosis creates 4 unique daughter cells with half the amount of DNA as the parent cell. So whenever you see the word or the phrase half of the amount of DNA, you know that you're talking about a haploid cell. So the question that we're going to answer throughout this activity is, how does meiosis create 4 daughter cells from one parent cell? So we're just going to walk right through it. So observe. Prophase 1, click on the nucleus to break it down, then click on the DNA to condense it into chromosomes. Drag the centrosomes to the top and bottom of the cell. So you're going to answer these questions as I do it. So I will come back to the gizmo after I get into metaphase and anaphase. So we need to click on the nucleus. We need to get rid of the nucleus. It needs to go away. Then we're going to click on our chromosomes, and they're starting to condense. Now we need to drag our centromeres to the opposite pole of the cell. So prophase 1. Then what's going to happen is the chromosomes are going to pair up with their homologous pair. So you can see we have a light green and a dark green, a light blue and a dark blue. They paired up with their homologous pair based on their size, their shape, their genes, the gene locations. Sorry. So now we're going to click continue. So now we're getting into metaphase. So our next step, observe metaphase 1 and anaphase 1. So remember, whenever there's a 1 after these phases, that means that we are in meiosis 1. We're going to drag the groups of homologous chromosomes to the metaphase plate, which is that middle plate. Then drag the spindle fibers from each of the centrosomes to the chromosomes. Click the centrosome to pull the chromosome apart. Here's a question here. How do the chromosomes separate in anaphase 1? So go ahead and watch. I'm going to move these chromosomes. Remember, they're in tetrads right now. Right along the metaphase plate, right in the middle. I'm going to bring some spindle fibers. Now there would be more, but just for the sake of the lab, there's only two, but there's a lot of spindle fibers, and they'll come through here and attach to each other. And remember, those spindle fibers are holding these in place. So now we're going into anaphase 1. Everything's being highlighted over here. Whoops. So they just split. So here's your question here that you need to answer. So compare. An image of the anaphase step in mitosis is shown to the right. Here's mitosis. You might need to blow it up so you can see it. I can do it here for you. Whoops. How does anaphase 1 in meiosis differ from anaphase in mitosis? So here's anaphase in mitosis, and here's anaphase in meiosis 1. All right. At the end of anaphase 1 in meiosis, how many chromosomes are on each side? Okay, so now we're going to go through, we're going to answer question 4, A and B. We're going to go through telophase 1 and cytokinesis. Those are the final steps in the first half of meiosis. You need to describe what happens when I click on the chromosomes during telophase 1, and then you need to, I'll click and drag on the contractile ring, which that is what's going to create our cleavage furrow. That's what's going to pinch off. Then you need to describe what happens during cytokinesis. So these two questions you can answer now. Come on. So as we can see, we just went through meiosis 1. We went from 1 cell to 2, and they finally split. All right, so go through the steps of the second half of meiosis until you reach the end of telophase 2. Following the instructions at the top right corner as you proceed, answer the questions below. Use the back button if you need to see a step again. So you're going to answer these questions right now. Before prophase 2 begins, does the DNA in the cell plate duplicate itself? Okay, so you're going to go through all these questions at this part right now. So in prophase 2, I'm going to condense my chromosomes. We're going to move the centrosomes to the opposite poles. We're going to line up our chromosomes. Remember, now they have 2 chromatids. In the middle of the cell, we're going to put our spindle fibers on those centromeres. Do the same thing up here. And then we're going to pull the chromatids apart in anaphase 2. So now we're at telophase 2. We're going to decondense those chromosomes back into chromatin. We're going to pinch off, create our cleavage furrow. Cytokinesis has occurred. So now these are called spermatids because, again, we are doing a male cell. All right, now let me see here. So now we're going to click on the spermatids to see the mature sperm cells. So this is what we talked about today in class. I drew 4 of these. They're the same size. They all look the same. The only thing that's different is the DNA inside, because remember, these cells are genetically different than the parent cell at the beginning of meiosis. So this is a male. All right, again, you can pause the video at any time. So I'll read this part for you. The original parent cell is called diploid because it contains a complete set of homologous chromosome pairs. Each of the 4 daughter cells is haploid, meaning that each contains half of the original parent cell's chromosomes. Each daughter cell contains 1 chromatid from each homologous pair. So right here it says click on the spermatids. Spermatids that formed from meiosis will develop into mature male gametes called sperm cells. Sketch a mature sperm cell to the right. So if you're just doing this on the paper, just sketch one of those. That's it. Doesn't need to be perfect. We're not artists. Mature sperm cells have only a small amount of cytoplasm, and they use their flagella, which, if you guys remember from the very beginning of the school year, this here is their flagella, or their tails, to propel themselves forward. Sperm are designed for one purpose, and that is to deliver genetic material to the egg cell during fertilization. All right, so if you have any questions as you're going through this lab tomorrow, please, please, please let me know. Okay, I will be available. So activity B, now we're going to do it for a female, I believe. Yes, although both male and female gametes contain genetic material from the parent organism, they perform different functions. A male gamete delivers genetic material to a female gamete. The fertilized female gamete, called a zygote, then grows into the offspring. So now during this activity, activity B, we're going to be answering this question. What are the differences in meiosis between male and female cells? So click on the female button. For the female cell, proceed through meiosis until you reach the end of anaphase 1. Up until this point, did you notice any differences between the development of a male and female gametes? So I'm going to reset. We're going to do female. I'm going to go through the whole way to anaphase 1. You need to answer this question. Whoops, this one here, sorry. That one there. All right, let's do it. So we're going to dissolve our nuclear membrane. Let's condense our chromosomes. Now we're going to, we have to move our centrosomes first. Now we're going to pair up our homologous chromosomes. Oh, here, I'll show you real quick. It says here we can make a crossover. So remember, prophase 1 is the only phase that crossing over exists. So what happens was they shook hands, so to speak, and they exchanged some genetic material between each other. So now we're going to line them up. Let's get those spindle fibers on those centromeres because we need to pull apart the tetrad. All right, so now we're on anaphase 1. So these colors here, they represent that crossing over. So they're going to be a little bit different. All right, so you should have answered that question. Now we're going to proceed through telophase 1 and cytokinesis. You're going to do these two questions now. So I'm just going to decondense our chromosomes, make them back into chromatin. Now take a look. Really observe these. All right. So now we're going to get into prophase 1 again. Or prophase 2. We're going to continue through meiosis until you finish telophase 2 and cytokinesis 2. So we're right here. So you're going to go ahead and answer these questions for this part here. So I'm going to decondense my chromosomes again. Let's move our centrosomes to opposite poles of the cell. We don't need to pair up our homologous chromosomes because they're all by themselves. So now we're going to add our spindle fibers during metaphase. Again, metaphase, they always line up in that equatorial plate right along the center. Whoops, if I can get it on there. All right, so now we're going to pull them apart. Same thing down here. Now we need to turn our chromosomes back into chromatin. We're going to decondense them. We're going to form new nuclei, and then we're going to split them. Okay, so we just went through cytokinesis. So this here is called the ovum, and this is the egg cell. These guys here, they're much smaller, and they're called polar bodies. So make sure that you're reading this information down here in the blue box as well. This is where you're going to get some of your answers. All right, so you should have went through here. So think and discuss. Why do you think the egg cells are large and sperm cells are small? So our egg cell right here. So even though we made 4 cells, this is the most important one. In females, what do they do that males don't do? Think of it that way. So why is this one so large and a sperm cell is so small? All right, so activity C. What we're going to do is we're going to answer this question for this activity. How can meiosis create an unlimited number of unique gametes? So I want to explain this to you because I don't want you to get confused. You're going to use the following abbreviations for the chromosome. So dark green, light green, dark purple, light purple. We can do male or female. It does not matter. So you're going to proceed through meiosis to anaphase 1. Which chromosomes went up and which ones went down? So I'm going to show you what I mean by that. So let's reset this. Let's just do a female. So we're going to go through. We went through interphase. The DNA copied himself. Now we're going to go through prophase 1. We're going to disintegrate that nucleus. And we're going to condense our chromosomes from chromatin. Let's move our centrosomes to the opposite poles. Let's line up our chromosomes. We're going to create tetrads. And let's have some crossing over. So we had some crossing over happen in the female egg cell that we're going to make. All right, whoops. So let's continue. Let's line up our homologous or our tetrad on the metaphase plate. Let's put our spindle fibers there to hold them in place and keep them lined up in the middle. Now let's pull the tetrad apart into their homologous chromosomes. So what they're saying here, proceed through meiosis to anaphase 1. So that's where we are right now. Which chromosomes went up and which went down? So now what they want us to do, so right now, for up, you should have these two. So dark green, dark blue. Down, light green, light blue. Okay? So now what they want us to do is they want us to click back and run anaphase 1 again for a few times. Did the results ever change? So remember, we had dark up here, light down here. So let's hit back. Darn it. And let's do it again. Did anything change? No. There's still, the dark green, dark blue, light green, and light blue down here. Let's try it again. I'm going to do it a few more times. Uh-oh. Look at that. Something changed. Now we have a light green up and a dark green down. Let's try it again. So what this part is just trying to tell you, which it sort of drives me nuts because it doesn't change all that often. But what this part is trying to tell you is that this is completely random. Remember I said in class, and I have repeated this and repeated this and repeated this. What goes on in this part here, pulling these chromosomes apart, is completely random. So it does change. So tell me why. Why do they change? So the chromosomes are distributed randomly during anaphase 1. What are the possible chromosome combinations in the two daughter cells? So again, you're just going to tell me which ones can go up, which ones can go down. This is sort of like a math problem from early junior high. So, dark green can pull a light green up. Dark green can also go up with a dark purple. Dark green can go up with a light purple. And then so on and so forth. So that's all you're doing there. All right, so click reset, choose a male or female cell, proceed through meiosis until the chromosomes are condensed in prophase 1. Drag the light green chromosome to the allele map on the left. This shows the alleles or the variations of a genes, and you're going to get into this next unit. You're getting into genetics that are present on the chromosome. A genotype is a list of alleles. So the genotype of the LG chromosome, for example, is, and then they give you an example. So what I'm going to do, I'm going to go ahead, we're just going to use a male one since I used a female one last time, it's not going to matter. We're going to proceed through meiosis until the chromosomes are condensed. So let's reset, male. Let's condense our chromosomes, get rid of our nucleus, and condense our chromosomes. All right, so what are the genotypes of the chromosome? So this is your chromosome for your light green. Or your genotype for your light green. Here's our genotype for our dark green. So let me take my dark green. You're going to write these letters. EE, FF, GG, HH, JJ. All lowercase. So that's what you're writing for the light green. That's what you're writing for the dark purple. And that is what you're writing for the light purple. All right, so after moving the centrosomes, drag the pair of homologous chromosomes together. Click on a chromosome. What happens? So let's go ahead and move our centrosomes. And let's click on a chromosome. What happens? Why did it do that? Why did this light green chromatid have a piece of dark green. Same thing over here with the purple. What happened? Remember, we're in prophase 1. This only happens in prophase 1. So when the homologous chromosomes are paired up, they can exchange sections. This exchange of genes is called crossing over. Click on several segments to create crossovers, and then click Continue. Proceed to anaphase 1, drag each chromosome to the allele map, and write a genotype. So I'll go ahead and do that. Alright, so continue. Let's line these suckers up on the metaphase plate. This is why I was really hoping this was going to work for you, because you're actually doing the chromosomes. You're moving them yourself. All right, so light green. Ready? Here's what you're writing. Make sure that you're writing them capital and lowercase. That's for light green. Dark green. Oops. Go back. Dark green. Light purple. Dark purple. Notice that they changed. They changed from up here. They're different now, and that's because of that crossing over. So think and discuss. In this gizmo, only one crossing over is allowed in each segment. In reality, crossovers can occur at almost any point along the chromosome. So what that means is whenever they're in prophase, they can occur all throughout the chromosome. It doesn't have to just be at the ends. This is just for the sake of easiness to make it easier. But they can occur all throughout. How do the random distribution of chromosomes and crossover create more variation in the resulting gametes? So you're going to answer that. All right, so meiosis is a complicated process. What happens when something goes wrong? Click reset and choose male or female cell. Click skip. Describe what would happen if meiosis occurred without DNA replication. So let's reset and click skip. So we're not going to copy our DNA. Now, if you watch, it's going to go through some of the phases. So this is what would happen if we were to skip DNA replication. So you need to tell me what would go on. What's happening? Why is this bad? Okay. Click back, proceed through meiosis until the chromosomes are lined up along the metaphase plate. So let's go back. So let's proceed. We're going to copy our DNA, disintegrate our nucleus, condense our chromosomes. And then we're going to move our centrosomes to the poles. Let's line up our chromosomes into their homologous pairs. Let's line these up at the metaphase plate. Click skip. What would happen if the chromosomes did not attach to the spindle fibers during metaphase 1? So I'm not going to attach my spindle fibers. We're going to skip. What's going to happen? So this is going to finish going through meiosis. So what's going on? Tell me what's happening. So click back, proceed through meiosis until the chromatids are connected to spindle fibers at anaphase 1. Click skip, describe what would happen if the sister chromatids were not pulled apart at anaphase 2. So I need to proceed to anaphase 2. So let's go ahead and attach those spindle fibers. Let's pull them apart. Let's decondense, or let's condense our chromosomes again. Let's move our centrosomes to the opposite poles because this is what's going to give us our spindle fibers. Okay. So let's see. Proceed through meiosis till the chromatids are connected to spindle fibers at anaphase 2. Click skip. Describe what would happen if the sister chromosomes were not pulled apart. So that's what you're doing. And again, if you read these boxes down here, the blue boxes, those are going to help you. So during meiosis, there are checkpoints that do stop cell division if anything goes wrong. However, these checks do not always work. Abnormal cell division during meiosis can lead to genetic disorders. Trisomy 21, and what this means is there is, oh, it's going to tell you, it's Down syndrome, but it occurs when there is an extra copy of chromosome 21 of the sex cells. All right, so that's it. That's all you're doing. There's a lot of pages here, but again, it's just because there is it's the instructions. So again, please, please, please let me know if you have any questions. And again, if this does work for you, please go in and do it yourself. You're going to learn a lot more if you're able to go in and do it yourself. But until then, I'm going to go ahead and go. Any questions, please get a hold of me tomorrow. My class tomorrow starts at 9:30. So I will have this up here. Well, you can't see it right here. So that way I can see the inbox. All right, so this video is going to go into this module. Remember, this is the new code. Please, remind yourself of that. And this is where you're going to submit it. If you have any problems, please let me know. I will be happy to help you. All right, guys, thanks so much. Make sure that you turn this in. This is due tomorrow at 11:59.